Okay, welcome and thank you for joining us for the Garvan Institute's annual From the Breast Cancer Lab to You Symposium. I'm Associate Professor Alex Swarbrick. I'm a lab head here at the Garvan Institute of Medical Research, and my lab sits within the Kinghorn Cancer Centre next door, which are both located in Darlinghurst in the centre of New South Wales. And I'm your MC for today. So the reason we're here is because, today in Australia alone, more than 40 women will be diagnosed with breast cancer. Now, 30 years ago, more than a quarter of these women would be expected to die of their disease within 5 years of diagnosis. Today, that number is far less. Less than 10% of breast cancer patients will die within the first 5 years after diagnosis, and many of them will go on to live perfectly healthy lives with their disease never recurring. And this progress, this incredible progress, has been made through research, through the advent of breast screening, but also through the advent of new diagnostic tests and new treatments. For example, Herceptin and tamoxifen, drugs that many of you may be familiar with and which have revolutionized the lives of millions of women around the world. Despite that progress, there's still many challenges to face in the area of breast cancer. So today, on average, for example, 9 Australian women will die of their breast cancer. Breast cancer affects younger people than other cancer types, and often these women are in their 40s, 50s, and 60s with many years of healthy life ahead of them and incredibly important connections to families, workplaces, friends, and so on. In fact, for women, breast cancer still steals more healthy years of life of any cancer. More than 62,000 person years of life are taken by breast cancer in 1 year in Australia. So today, we're going to hear about the latest in breast cancer research and medicine and hear a personal story from a breast cancer patient. We're going to encourage you to ask questions, and you can do that by clicking the Q&A button at the bottom of your Zoom window and type your question. If you're interested in a question that's already been asked, you can upvote it, so you can click like, and that question will go to the top of the list. At the end of today's session, we'll get to some of those questions. Unfortunately, we won't have time to deal with all of them, but we'll do our best to answer those questions for you. Now, before we hear from our wonderful speakers today, I'd like to pay my respects to the Gadigal people of the Eora Nation as the traditional owners and custodians of the land on which the Garvan Institute, the Kinghorn Cancer Centre, and the St Vincent's Hospital are located. We recognise their continuing connection to land, waters, and culture, and pay our respects to their elders past, present, and emerging. So really the first order of business for today is to thank Estee Lauder, our sponsor, for their really wonderful support of this event. And they've been supporting this event for a number of years now. And this has come through the Estee Lauder Company's breast cancer campaign, which is something we're extremely grateful for. This year, we're announcing a research award funded by Estee Lauder to help fund an innovative new project in breast cancer research, kind of seed funding for a project that we hope will blossom into a much larger, substantial project that can then recruit competitive peer-reviewed funding from major funding bodies. So to say a few words about Estee Lauder's support of this event, I'd like to introduce Emerentia Wilding, who's the General Manager of Estee Lauder Companies Australia and New Zealand.
Thank you, Alex. 2020 has indeed been a year like none of us have ever experienced, and we recognise that people around the world are facing different health crises every day, each deserving attention and support. But our long-term commitment to defeat breast cancer will not falter. For 28 years, the Estee Lauder Companies has been committed to our mission to create a breast cancer-free world. Our campaign began when Mrs. Evelyn Lauder co-created the Pink Ribbon in 1992 to bring a level of global awareness to this disease through the collective voices of our employees, our partners, and our customers. Today, our commitment to this cause is as strong as ever, and I'm really pleased to report that globally, we have raised over $89 million for life-saving research, education, and medical services since the campaign began. We are truly honoured to support, for the 4th consecutive year, the Garvan Institute through our sponsorship of the Breast Cancer Symposium. We believe that the opportunity to engage with the community and highlight the groundbreaking work of their team helps to unite us in our collective efforts to raise awareness, inspire action, and ultimately find a cure. Inspired by the work of Garvan, the Estee Lauder employees in Australia continue to come together to raise funds, and it is through their efforts that we're able to support early-stage breast cancer research. Funding early-stage research is critical for PhD students and others starting out on their research careers, and receiving this financial support enables them to gather preliminary scientific research evidence needed to apply for competitive peer-reviewed funding and government grants. So with this in mind, I'm honoured to present this award to Sarah Alexandru, and on behalf of all at Estee Lauder companies, I want to congratulate Sarah and thank her for her incredible work.
Thank you so much, Amarentia, and congratulations, Sarah. It's a really wonderful award. I know she's very excited. She's unable to join us today to accept that award, but on behalf of Sarah and all the breast cancer researchers here at Garvan, we're really thankful to you for supporting this event, but also supporting Sarah's really exciting new project. She's a PhD student in Liz Calvin's group here at Garvan, and we'll be hearing a little more from Liz Calvin a little later on. So thank you very much, and thank you, Estee Lauder. Okay, so what we'd like to do now is to meet someone with a very close understanding of breast cancer and its treatment. And in fact, we're going to meet someone who's a breast cancer patient being treated here at the Kinghorn Cancer Centre. I'm really pleased to introduce Kate McDonald, who's very kindly agreed to come and to tell us about her breast cancer experience. So Kate, welcome to the panel.
Hello, Alex, thank you.
You're very welcome. So,
sorry,
hi.
Alex, I'm just having trouble just hearing you.
Oh, you are? Okay. How's that going? Is that all right? Can you hear me at all?
Sorry. Yes, I can hear you now. Sorry. Yes.
Okay. We will never master this Zoom thing. Trust me. So thank you so much for joining us today. It's really a great honor and a pleasure to have you here. So as I was just mentioning, you are a patient here at the Kinghorn Cancer Centre, breast cancer patient. And so we'd really love to kind of hear from you, your perspective on your journey. So I guess to start, maybe if you could tell us a little about yourself, what do you do and how do you find yourself here on this panel?
Yes, so I born and bred in Sydney. I'm a journalist. I work on a radio program. I produce a radio program which deals in international politics and foreign affairs. So very different to this panel. And so I was diagnosed last May with stage 3 breast cancer. I just went for a routine checkup. I've been having them every 2 years since I turned 40. Not because there was any breast cancer in my family. It was my father had bowel cancer, so I started doing bowel checks at the age of 40. So my GP at the time thought it was a good idea to also start getting having mammograms. And so it was just a routine checkup, no idea whatsoever that I had a lump. And even after the diagnosis, I still couldn't feel the lump because it was quite small. And you know, I just think that screening is so important. And it's great because lots of my friends now are getting screening at last. And so I had a lumpectomy. And it was when they did that, the pathology came back and they realized it was a lot more aggressive than what they had first thought. I think 12 out of my 16 lymph nodes had cancer in it. Feisty is how my oncologist called it, you know, small but aggressive. And so I had so after the lumpectomy, I then did 5 months of chemo. So it was 16 rounds in 20 weeks. I'm not very good with the the drug names, but I did the red poison I had 4 rounds of red poison and 12 rounds of what I called the yucky you. But I coped quite well with the chemotherapy, which I also attribute very much to my oncologist as well, who helped me through all that. So fatigue was that was the biggest killer for me. I was just so tired with it, which was a good excuse to watch really bad TV on the lounge and eat chocolate ice cream. I also didn't lose my taste at all. I just, I actually put on 5 kilos during chemo. So, and then I had the, 3 weeks after chemo, I had the mastectomy. So I had the right breast taken off. That was then followed by, I think, 28 rounds of radiotherapy, which I think is the maximum that you can have. I also started on an anti-hormonal drug, letrozole. So I started on that at the end of December. So I think I'm on that for about 5 years or so. So it's been a very kind of long road, but you feel it, you know, incredibly supported by all the medical team, and that really gets you through. And I think that's a really important thing, to have a medical team that you really trust as well. And it's not saying that you don't go and ask lots of questions. I think all of my medical team probably hated me coming in with my notepad and a pen. That's a bit of a professional kind of, you know, hazard, I guess. But it's that trust is really important, and I trusted them, and yeah, so that's the journey that I had with the which I'm still on, I guess, that cancer journey, the treatment.
Right. And of course, you know, the last 8 months or so have been incredibly difficult times for everyone, but it's kind of hard to imagine going through what you just shared with us in the times of COVID-19. So how has the pandemic, you know, it must have really impacted not just your kind of emotional ability to kind of deal with what you were going through, but also the ability for you to receive medical care. How have you seen that?
Well, I actually think I was extremely lucky, because I had just finished radiotherapy as COVID was kind of, you know, coming to Sydney or coming to Australia. So that was the beginning of March. And I think it must be so difficult for women to be going through it now, like not to have that support, because last year during chemo, I just had, you know, endless visitors coming and bringing food and just coming and just talking. It was an extremely busy period of my life, actually. And I just think this year it must be very difficult. It's, I've had, you know, a couple of telehealth meetings, but I have been able to come in to like the Kinghorn Cancer Centre for those meetings with the mask on. The mask is actually quite interesting. I had a bone scan last week, a total bone scan, which I had at the beginning of this process last year. And I was fine with it last year. I think wearing the mask and having that thing so close to your face, that really did freak me out. So again, I just think, you know, I was very lucky that I got through most of that treatment without all the restrictions of COVID.
Right. Thanks for sharing that. So you mentioned that you're still receiving treatment, and from what I understand, that's part of a clinical trial. So you're part of a study exploring a new potential treatment for breast cancer. Would you mind telling me a little bit about the trial that you're on and why you chose to participate in a clinical trial?
So the trial is for a drug which I hope I can pronounce correctly, ribociclib, which is, now I've got a trial for women with high-risk hormone receptor breast cancer, which is me. It's an international trial. I think there's about 4,000 women who are participating. And when my oncologist approached me about it last year, I have to admit, at first I was thinking, oh, no way, because you look at all the side effects, and after just coming through chemo, it was like, I just don't know if I can put my body through this again. But I have to say the side effects have just been basically nil, I think. And you just, with these trials, you just know that I was fascinated last year when I was going through all this about all those people who first had like the red poison drug that I had, and the people who first had an MRI, and just how much they, you know, put themselves out so that people like me years later can have the benefit of, you know, all that research, and for them being the first people to do it. So it actually ended up being a no-brainer. Like, of course, you're going to put your hand up to participate so that, you know, other people coming through can have an easier ride like people have done, you know, 20 years ago. And you also think about Connie Johnson as well, and, you know, just what an incredible legacy, you know, that the breast research group and her family and everything that they're doing for people like me as well.
Oh, that's a brilliant answer. Thank you, Kate. I know that you're doing more than just participating in a clinical trial. You're also sharing some of your cancer tissue with researchers. So here at Garvan, a lot of the research we do is patient-focused. It's actually studying people. And at the center of that is finding patients who are willing to be partners in that research. And so this is a program that we call Project Share. And I believe you're a participant in Project Share. So what kind of motivated you to join in that kind of tissue donation program, and what's your experience been of it? Have you found it to be a kind of a positive thing, a negative thing?
Oh, to tell you the truth, I didn't want that cancer tissue, so it's like I'm very happy to hand it over. So that was, again, that was just, it was actually quite an easy decision. Again, it's, you know, and, you know, it hasn't, you know, really impacted on me at all, that part of, you know, giving the cancer tissue. And again, it's that thing to, like, whatever can be done to try and help and to, you know, to get the research and to try, you know, and have, you know, enable people to have longer and healthier lives. And I probably shouldn't be saying this for so many people listening, but I'm very happy to continue to participate in any trials that can help future patients.
Right. Well, Kate, I really want to thank you for not only participating in our clinical research, in our lab-based research here at Garvan, but also coming today to share your story and being so open with us. I really thank you for that. And I hope everybody watching at home has, I think, probably learned a lot from your perspective. So thank you very much.
Okay, thank you, Alex.
Okay, well, that was really wonderful hearing from Kate, the kind of inside story on, you know, what it's like to be a breast cancer patient. Now I'd like to introduce to you Associate Professor Elgene Lim, who is both a lab head and scientist here at the Garvan Institute, but also a medical oncologist. So he treats patients like Kate in the clinic every day. And Elgene is going to take over here talking about some of the cutting edge research and medicine in breast cancer. So Elgene, over to you.
Okay, so this particular segment, I will be introducing the winner of this year's Connie Johnson Memorial Lecture Award. And it gives me, which is this, it gives me great pleasure to introduce Dr. Sarat Chandralapati as this year's recipient of the Connie Johnson Memorial Lecture Award. And this award is given out each year to an outstanding international breast cancer researcher. So Sarat is an internationally leading medical oncologist and laboratory head at the Memorial Sloan Kettering Cancer Center in New York City. A major focus of his work has been to characterize the significance of changes that are found in cancers that are spread to other parts of the body and that have also become resistant to the current gold standard therapies that we currently use. He has been a Breast Cancer Research Foundation and Estee Lauder funded researcher since 2015. So we're in for a treat today, and thank you so much, Sarat, no pressure. But before Sarat actually kicks off, I'd like to invite Hilda to speak a few words. Hilda is an author and sister of Connie Johnson, OA, in whom this lectureship is named after. Hilda will be reflecting on behalf of the Johnson family of Connie's legacy and her continued inspiration to us all. So thank you very much, Hilda.
Oh, thank you, Elgene. Nice to see you, albeit on screen. Yeah, so I'm Hilda. I'm the older sister of Connie and Sam Johnson, and we started our charity Love Your Sister when Connie got diagnosed with terminal cancer. And so far we've raised $13 million for varying research projects, and we're very proud of that. But The establishment of the Connie Johnson Lab was one of Con's proudest moments, and is still one of mine. I had the opportunity to visit the lab with her husband Mike and their boys a couple of years ago, and it was an incredibly emotional experience being there, seeing her legacy in bricks and mortar. Yeah, it's very emotional for me. So the sort of character of Connie really shone through. I'll focus particularly sort of at her end of life. In the last 5 days, the Governor-General rang up and wanted to award her an OAM for all her work into research and fundraising. And part of that process was moving the cars in the car park. And Connie declined the award because she didn't want to inconvenience other patients. Needless to say, they decided not to move the cars and did it anyway, but I think that sort of really speaks to her character. The other thing she was very keen on was having a warm autopsy, which Garvan was involved in. So when Connie died, I had the very important job of calling Elgene straight away, and Connie was taken to the Garvan lab so they could harvest the live cancer tissue cells and do all the sciencey things. It's not my area. Yeah, so anything to do with fundraising and research is her legacy, and we're proud to continue it. And this Memorial Award is just a reminder of who she was and what she wanted to do. And it means a great deal to our family. And we promise to keep working as hard as you do. Thanks.
Thank you very much, Hilda. Over to you, Surat.
All right. Well, let me begin by thanking you very much for this incredible honor. I, of course, never had the privilege of meeting Connie, but I am a practicing oncologist like Elgene. And even today, you know, I had to speak to a patient of mine to tell her that a therapy failed her. You know, I had the same sort of questions that she had. Why did that happen? And what can we do about it? What's the science behind why that happened? And what's scientifically proven that we can do about it? So it's my privilege really to have a laboratory that seeks to address those very questions about why our treatments fail our patients and how we can do better. And so in my talk today, you know, I want to really address where we are in this journey of trying to sort of develop new therapies based on our understanding of how cancers become resistant. Sort of how, what I've titled is How Well-Behaved Therapies Adapt, how well-behaved tumors adapt to therapy, and how we are adapting our therapies to defeat them. I'll begin with a kind of obvious question, and a simple question, but an obvious one. Why do patients die of breast cancer? I mean, so the breast is not a vital organ. It's not the lungs or the brain. So what is the cause of death? And here I show the epidemiology, which I think gives a bit of a clue. Shown here are 5-year survival rates for patients with breast cancer that's localized to the breast, or localized to the breast and lymph nodes, or spread distantly to other organs like the bone or liver. And really, you know, I think it's pretty clear that there are differences here once the cancer is spread to distant organs. Now, why is it that the cancers that are localized are so much better treated or cured than those that are distant? It's breast cancer either way. and in fact, we offer many of the same treatments: surgery, radiation, chemotherapies, hormone therapies, but it's clearly much more successful in the case of localized disease. So why is that? And what I want to suggest is it's not just the location of the cancer, that it's something more than that. And while the location is important, what I think is also evident is that the cancers are evolving and changing as they go from a localized to a distant disease. And this goes back to work from Peter Knoll, many decades ago, where in studying cancer, he demonstrated that the cancers, as they grow over time, they become more diverse. They start off in this example as just gray with one founder cell, but as they grow, they become this collection of related clones, related populations of cancer. That process is even more the case for cancers that have spread to distant organs. At least that was our hypothesis. And going beyond that, what does that complexity involve? What we speculated was that complexity involved clones of cancers that become resistant to the therapies we're giving. So to study that, to understand that, we went to the most commonly used therapy in breast cancer, which is hormonal therapy. You heard that term, letrozole, earlier, anti-estrogen therapy. And again, estrogens are fundamental to the mammary gland, as shown here in this mouse or rat that had normal mammary gland and no estrogen, no mammary gland. And anti-estrogens are among the most effective therapies in cancer today. In fact, I would say in the history of cancer medicine, anti-estrogens might have saved more lives through their use in early stage cancer than any other therapy. But in the advanced distant setting, we do see that therapies work more transiently, maybe for a couple of years, as shown in this curve where patients are coming off of therapy because it's failed them. Why does it fail? That was the question we have. Why does it fail in this distant setting where it so less often does in the early setting? And so what we did was we asked of our patients, could we collect tissue, just like Connie donated tissue, after the cancer had progressed or not responded to the hormone therapy, and compare that to early breast cancers. And what we found in this rather colorful diagram and picture here was that there were mutations that were present in the cancer that had spread that was not present in the primary early cancer. What do I mean by that? So what we did was we sequenced, just like we can sequence DNA for the whole genome, like was done in the 1990s for one patient. Now we can do that at scale for individual cancer patients to look at their cancer genome. We can study what mutations are present in the DNA, and we can do that both in the early cancer as well as the later cancer. And what we found is in the later cancer, there were new mutations. The cancer had evolved. New clones had emerged, so to speak. That red color came out from the gray color. And those mutations actually caused the cancer to not respond or not need estrogen anymore. They became hormone resistant. They became resistant to all of our anti-estrogen therapies. And there were a number of mutations like that. It wasn't just anti-estrogens. We've studied this now with many therapies used in breast cancer, and what we find is the same, that after cancers have been exposed to therapies and in distant sites, they acquire new mutations that render them resistant to the therapy that we gave. They evolved, they changed. the original cancer didn't have these mutations and responded, would be predicted, responded quite well and do. But what we're seeing in these curves and these graphs is cancers that didn't have mutations at the beginning and then developed them over time. So what we have, I think, with distant disease is a problem not merely of location, but we have a problem of cancer evolution, of an adaptable organism, so to speak. And so how do we deal with that problem? What I would say is the problem is quite challenging because we can't simply just treat that one resistant mutation. We tried that. In fact, that was the first obvious thing that we did. We found, for instance, that mutation in the estrogen receptor gene. We developed a drug against that. The problem, however, was the cancer then evolved to something else. It evolved another mutation, and it in fact has the capacity to evolve several. So what we were dealing with was, I think, a more challenging problem than that. And so what are the solutions that we've started to work through? And I would say this is, many groups doing it, but this is, these are representing, I think, 3 areas that we think are quite promising in terms of a strategy to deal with this problem of cancer evolution. The first is early detection. It goes back to this very idea that that if you can get the cancer a little bit earlier, it hasn't had time to become more complex. And so how do we do that? Well, one of the ways that's been developed by the technology, the ability to sequence, is to look at the DNA that is released by the cancer into the bloodstream. So we know that cancer cells, because they're dividing more often, they release their DNA into the bloodstream, and we can pick up the mutations with a blood test. And with increasing sensitivity, we're actually able to find these mutations. And we've been working very hard on the technology to be able to do a blood test so that we can find these mutations. And we actually can track mutations over time. So that ability to serially monitor a patient may allow us to detect a cancer much earlier and see this process of evolution perhaps in real time. And this is just one depiction of an example. This is a patient from the ICR who had their circulating tumor DNA blood tests looking for mutations. And what they found remarkably was that the mutations preceded detection of that distant disease, in this case in the liver, a full 13 months before the imaging did. So for a full year, this cancer was evolving and growing before they ever could detect it using the standard technologies. And so this, this was all done retrospectively, but the point is that you can, with this technology, you may be able to detect things far earlier than we can currently, waiting for imaging to show what's going on. And that perhaps gives us the opportunity to intervene earlier, just like the hormone therapy or the chemotherapy being given earlier can make them more effective because the cancer hasn't evolved as much. In the same way, if we can detect things earlier, we can introduce our newer treatments earlier in the course of the disease. And we are doing precisely that in clinical trials today. We are we are essentially finding these patients who have just what we call a molecular relapse or molecular evidence of cancer, and then introducing a drug at that point rather than waiting for the radiographic demonstration. And so this technology, I think, is very promising, and it's being used by many of us to try to help identify patients earlier. The second is to understand this process of evolution. What is actually allowing these new mutations to show up? So if you look at patient with lung cancer, and you look for the mutations present there, and you put them together in aggregate, what you find is sort of a signature of smoking. And if you look at a patient with melanoma, and you look at all those mutations in aggregate, what you find is a signature of UV sunlight. What about in breast cancer? Well, in breast cancer, we don't have such a signature like smoking or, tobacco or UV sunlight. What we have are some other signatures, and this is work that many of us are doing, is to find the signatures. And what is remarkable is when we look in patients with that distant spread disease, what we find is an increasing number with a signature called ApoBec. And ApoBec is representing a family of normal enzymes that our cells make that might be used in viral defense, but are being co-opted here to introduce new mutations in the cancer and increase its complexity and potentially promote resistance. And that's what we've been finding increasingly in patients with distant spread disease. And so what we are now working on are drugs that can target ApoBec. Can we block essentially the evolutionary process that's going on? And there are several approaches to target evolution of the cancer, whether that's the ApoBec process or whether that's BRCA1 and 2 defects. Those are different evolutionary processes which we might be able to attack, and so prevent the source of these new mutations and the diversity of the cancer from expanding. And then finally, we've been looking at approaches to try to increase the effect of our early treatments. And you heard about a clinical trial approach of one such strategy, and that's with the ribociclib. What we're trying to do, I think, is really try to make the localized cancer respond even more powerfully to our therapies. So anti-estrogens, what they do to a cancer cell is they make it arrest. They make it just stop dividing. And they put it into this sort of dormant state where it's quiet, it doesn't grow, but it only does that for a time, and eventually it could potentially grow out, particularly if there are a lot of cells. And so what we're trying to do is to convert that dormant cell into one that will never grow. It's in this case called senescence. So you might think of senescence as, well, like at the top of my head, I have hairs that just they're hair follicles that are never going to make any more hair, and it's just nothing I can do to change that. It's the same way. Can we make those rare cancer cells out there not just dormant but senescent? Put them into a state that they can never grow again. And there are actually drugs that we see in the lab, and Elgene's working on them, and we are as well, that can actually sort of induce that state, that permanent arrest state, and represent, I think, a way of making our early stage therapies even more effective, so that distant disease never emerges. And so this is a third approach I think that we're all trying to do, of trying to essentially really eradicate any distant disease that might be lurking. So I hope that I've at least been able to share with you some of the ideas behind how we are trying to improve upon our therapies and really eliminate the sort of scourge of distant and resistant and evolving breast cancer. I think, again, I would just want to highlight that these represent some of the approaches. There are others, of course, but I think that we are already seeing the first fruits of this in some of the clinical trials that we and others are participating. And so with that, I'll just close by thanking and acknowledging that this is work of many, many people, my laboratory, many collaborators, both academic as well as pharmaceutical, You know, it is really my privilege to be able to do this kind of work on behalf of my patients and all the patients who you know, are dealing with this dreaded disease. So I thank you and look forward to, I guess, the Q&A session after.
Sarat, thank you so much for that really brilliant overview of your work, really provocative and exciting. There's so many new things happening in this space. And of course, Sarat is really at the leading edge of this. And thank you very much for dialing in from all the way from New York City at night. We really appreciate it. We won't have questions now, but Sarat's going to hang around and we'll go to some questions that you're very welcome to put into the Q&A box for us to finish up on. So thank you very much. Okay, so I'd like to change now to give everyone an opportunity to learn a little bit about some of the work that's happening more locally and specifically here at the Garvan Institute. So we're really lucky to have my colleague, Liz Calden, who's another lab head here at the Garvan Institute, who's kindly agreed to give an overview of some of the cool new breast cancer research that's happening on campus. So Liz, over to you. Thank you.
Hello, hopefully I'm coming on screen now and you can hear me.
Yeah, all good.
All good? Great. So I'm Dr. Liz Calden. I'm going to put my slides up now and share them with you. And I'm now going to tell you about the breast cancer research that we are undertaking here at the Garvan Institute. It's a real privilege to represent all the breast cancer researchers here at the Garvan. There's in fact about 12 different groups working on breast cancer and about 50 different researchers working on breast cancer at the Garvan. So it's a really phenomenal effort we've got going here. And I've given this talk for the last couple of years and it gives me an opportunity to review what's happened in the last year. It's been a challenging year because of COVID-19, but I've got to say it's also been an extraordinarily productive year from our team working in breast cancer. And I'm really pleased to be able to tell you some of that information today. So we've heard a little bit about how, from when Alex did his introduction, about just how bad breast cancer continues to be. So it is still the most diagnosed cancer and it leads to more than 3,000 deaths in Australia every year. And of course, a lot more worldwide. And if you look at this schematic of patients, what you can see is that if you think about yourself, you may of course be a breast cancer sufferer or survivor yourself, but if you're not, you think about the people around you, you are going to know someone who has breast cancer, or more likely several people who have breast cancer in their lifetime. And in fact, 13 out of every 100 women do get breast cancer in their lifetime. But what research has done, which is truly extraordinary, is it's changed what happens to those people. So we now find that 5 years after diagnosis, instead of being the way it was back in 1930, where only a handful of those people would still be alive 5 years following diagnosis, we now find that the majority of those people are alive 5 years following their diagnosis. And what this means is that as researchers, we can now really focus on how can we actually treat these people better. So instead of just looking at numbers, we've really moved more to looking at to thinking about people when we think about breast cancer. And in fact, we can think about what each individual's cancer is like and what are the needs of that individual patient, rather than just looking at breast cancer as a very homogeneous problem. And part of what we look at in this process as researchers is the patient journey, because there's lots of stages in the patient journey where we can intervene and make an impact with research. So we can actually make an impact thinking about how did the cancer first start, what type of breast cancer is it, what drugs can we use to best treat that particular type of breast cancer, and then how can we best use that particular drug, and then finally, what treatments and what screenings can we use to screen breast cancer to make sure it doesn't come back. So what I'd like to do now is just tell you a little bit about the research from the different labs. I don't have a long period of time, so it's pretty broad brushstrokes the way I'm going to be describing this research to you. But what I hope is be able to give you some key messages from the research that's been going on at the Garvan. I'd like to start by first telling you about the research of Dr. Joanna Ashinga Koecker, who's part of Professor Sue Clark's epigenetics group at the Garvan. And I want to tell you about Jo's research because she's the Estee Lauder winner from 2020. So what Jo studies is she studies the way that DNA is organized in a cancer cell. And what you can see from this diagram, and I'm not having my screen update properly here, so I really do hope you are seeing all my slides. But what you can see from this diagram is that DNA, here represented as a purple string, is really well organized within a normal cell. It's actually very tightly bundled in these little structures that you can see here. And this organization is a fundamental aspect of how a normal cell does its job. And we often think of it as scientists as being little balls of string. And that really tight organization is important because each little bit of organization, each little bit of information is given in the right context. But what's really interesting about a cancer cell is that a cancer cell has extremely disorganized DNA, where we find that each piece of information is actually put in the wrong context, often giving the wrong information at the wrong time. So cells might be given the instructions to grow at the wrong time or invade at the wrong time, when in fact they should not be doing any of those activities. So Joanna's research has been continuing in this area this year in 2020, culminating in a really lovely publication that's come out this year. And Jo and Sue's group are now continuing in this area, really looking at how they might in the future reverse those changes in a cancer cell. I'd like to now move over and focus on the work of our MC today, Alex Swarbrick, who is doing some fantastic work looking at the heterogeneity in breast cancers. So often we think of breast cancer as just being kind of a lump that forms and assume everything's the same within that lump, but that's actually not true. Each cancer is made up of lots and lots of different cells. And using this incredible technique that Alex is using called single cell sequencing, he's pulling those cancers apart and looking at what those cancers are actually made up of. And when he does that, what he and his group have been able to show is that there's actually loads and loads of different types of cells within those cancers, and they can each have different functions in the cancer. In fact, they've discovered a new cell type which they think might be extremely important in cancers, which they've published this year as well. Likewise, Associate Professor Christine Schaffer is studying what's going on with different types of cells in cancers. And she works on a small group of cancers, which are a small group of cells, I should say, breast cancer cells in breast cancers, that are very highly adaptable cells, which otherwise known as very plastic cells, meaning they can take on different identities and different functions. And what she's showing is that these cells have this incredible ability to invade and avoid therapy. Now, this is important when we think about breast cancer and how the disease progresses, because when we think about the disease, it's not just about how that lump forms in the breast, it's also about how those cells actually then travel into the bloodstream and set up in another site in the body. Because as Surat has told us, it's really metastasis that can end up killing breast cancer patients. So we need to understand what it is about breast cancer that makes it move to another site in the body. Surat's also told us a lot about hormone-sensitive breast cancers, which make up the majority of breast cancers. And these have been indeed very successfully treated with long-term hormone therapy. And in fact, there's probably about 60,000 survivors of hormone receptor positive breast cancer in Australia today. Unfortunately, hormone receptor positive breast cancer, while very successfully treated, it actually has quite a high rate of recurrence in about 30% of patients. And this is really where a lot of our research is heading towards, to understanding what's going on here. Professor Chris Ormandy is trying to improve the way we can actually study the disease by creating new models of the disease. In the past, it's been notoriously difficult to study. And myself and Associate Professor Elgene Lim are working on really understanding what happens in very advanced disease, which is estrogen receptor positive. This disease is treated with lots and lots of different drugs. And as Surat has told us, those drugs change the cancer. So we're trying to create models of the cancer as it's been changed, so we understand what's going on in those newly changed cancer cells, and then generate better therapies for those newly changed cancer cells. So developing new treatments is a really important focus of what we do here at the Garvan Institute, and we've got a number of groups working in this area. We've got Associate Professor Tom Cox, who works on the stiffness in breast cancers. So as you know, they occur as lumps, but that lumpiness and that hardness is a really intrinsic factor about a cancer. And he's working on therapies which target that hardness and that lumpiness to actually see if that can be effective therapeutically. Associate Professor David Croucher is working on what are the differences between the primary disease and the metastatic disease, and whether we can use different drugs to treat metastatic disease instead of primary disease. And of course, there's lots of other cancers out there, and can we use the knowledge in those cancers to help us better treat breast cancers? And Dr. Simon Janunka is working on immunotherapy, which has been very successful in cancers such as melanoma, but not in breast cancer. And he has a great project looking at how do we actually know, what's the reason it doesn't work well in breast cancers, and how can we actually apply immunotherapy effectively in breast cancers? And finally, there's a lot of treatments out there, as many of you in the audience will know, as breast cancer patients and breast cancer survivors. But how can we actually make those particular therapies better? Associate Professor Claire Sturzaker is working on identifying that group of patients which actually responds best to the therapy and those who don't. And in that way, we can avoid giving therapy to those patients who won't actually benefit from it. And she's doing this through epigenetic signatures which occur in those patients. And finally, Dr. Max Nobis and a number of other researchers are looking at how do we make drugs work better by using them in new combinations or with different timing. And in that way, can we actually stop recurrences because we've actually used drugs more effectively in the first place? So what's the outcome of all of this research? Well, I've mentioned already that we've had a number of publications, and I was really proud when I reviewed this just to see how many different papers have been produced by all the groups across the Garvan this year. It's really astounding, and I applaud my colleagues for all their hard work. But really, one of our most important outcomes is how do we take this forward into patients? And that's where Associate Professor Elgene Lim is working really hard, looking at how do we implement our findings and take them forward into the clinic. And that's an area that he's going to tell us about next. But just before I finish up speaking to you, I'd just like to say a really important aspect of what we do is how we make it happen. And that's through funding from philanthropic organisations and from government agencies. We work very hard fundraising as well to try to make sure we can continue this valuable research, and these organisations have all given us really invaluable support. But of course, the patients who commit to participating in clinical trials like Kate, and also all of our other supporters, your support is pivotal. It really gives us the energy and it gives us the passion to keep doing what we're doing. So thank you very much.
Thank you so much, Liz. That is very impressive overview. It's a really difficult task summarizing the work of 12 groups, probably 100 researchers in total here at Garvan or more working on breast cancer research. So Liz has done a brilliant job in distilling that. So thank you very much. I'd like to now return to Associate Professor Elgene Lim, who's going to wear his doctor's hat, and he's going to update us on some of the latest activities in medical activities in breast cancer treatment ongoing here at the St. Vincent's campus.
Great, thanks, Alex. So I will be talking about some of the clinical trials at the Kinghorn Cancer Centre at St. Vincent's Hospital. But firstly, for those who are not familiar with clinical trials, these are a type of research that studies new tests and treatments and evaluates their effects on human health outcomes. It can range from a first-in-human study of new treatments to treatments which are compared to the current standards of care. And we have a very large clinical breast cancer trial program at the Kinghorn Cancer Centre, where patients partner with us, patients such as Kate partner with us to improve treatments and patient outcomes. And it includes investigator-led academic trials, as well as pharmaceutical-sponsored trials. So I've tried to capture some of these trials pictorially. A lot of our research has been on the hormone receptor positive breast cancer. And we've talked about tamoxifen and drugs such as letrozole, but there are new drugs that are potentially even more effective, called selective estrogen receptor degraders. And what it does is it breaks down that estrogen receptor that sits inside the cancer cell. So there are a number of these coming through. Importantly, they're oral, so they can be taken as a tablet. And we're fortunate to have a number of these drugs being evaluated at the Kinghorn. In addition, a cousin of the estrogen receptor is the progesterone receptor. And based on seminal work coming from an Australian in Cambridge, Jason Carroll, we've been able to start a clinical trial looking at the effect of progestins that activate the progesterone receptor in breast cancer. And this is a national trial where we're coordinating that trial. We've also talked about the CDK4/6 inhibitors, which has been used very effectively to impact the cell cycle progression of the cancer cells. And this is often given in combination with anti-hormone therapies. So while it's been shown to be very effective in patients with advanced breast cancer, a lot of our trials now are trying to evaluate whether this treatment is also effective in patients with early stage breast cancer. And Kate, our patient earlier on, also spoke about this trial. In the triple negative space, we have a lot of trials on immunotherapies. So immunotherapies are treatments that stimulate our immune system to better attack the cancer cell. We've also got cancer vaccine trials, which also aimed at doing the same thing about enhancing our immune response towards the cancer. We have a genomic medicine program. Sarat talked about testing the cancer DNA in the blood or in the tumor. And from that, we can then direct the patient treatment according to the mutations that are present in that individual's tumor. Another class of drugs that we are evaluating are antibody drug conjugates, and I call this the guided missile. We have the antibody, which takes the chemotherapy, which are these little yellow dots, specifically to cancer cells that have a particular target, and these antibody drug conjugates get internalized and the chemotherapy gets released within the cancer cell. So there is very little bystander effect, which is a problem with traditional chemotherapy. We've also got other, not therapeutic trials, but other type of trials happening, including a prevention trial for patients with BRCA1 mutations. We've got supportive care studies, for example, with vaginal dryness, which is very common with our anti-hormone therapies. We've got studies to evaluate treatments for that. We also have an exercise study that recently concluded in women with early stage breast cancer undergoing chemotherapy, evaluating the feasibility of going through a supervised exercise program concurrently with chemotherapy, and that was a great success. And then finally, we've got novel imaging studies trying to find better ways of imaging breast cancer in the body. As mentioned before, I think a key part of all our research here is the participation of patients. And this underpins a lot of our translational research here at the Garvan and Kinghorn, where we invite patients to share their specimens, as it will help all research efforts. And we ask women to donate not just cancer tissue from the breast, we also encourage women to give us a piece of their normal breast should they be undergoing a mastectomy operation, or if they have cancer that has spread to other parts of the body. Many women have partnered with us by giving us a biopsy of those samples for research. Finally, we are developing a clinical trials navigator. As we have this explosion of clinical trials in Australia, it's very difficult to navigate to find the right trial for you. So what we've done is work together with Optus, National Breast Cancer Foundation, Love Your Sister, to develop a patient-centric platform where patients can actually enter their details and be directed to a clinical trial that is relevant to them. And this empowers patients to be able to navigate a very complicated clinical trial environment that is emerging in Australia. It also helps, we hope, connect patients directly to researchers in Australia that are working on their type of breast cancer and facilitating research that way. So finally, on behalf of all the researchers at the Garvan and all the clinicians at St. Vincent's Hospital, I'd like to thank you all, patients and your families who have partnered with us in our quest to improve the outcomes of patients with breast cancer. If you're interested in the trials that we're running here, that's the website to look at. And if you're interested in some of the research that's happening in breast cancer at the Garvan, that's the website. Thank you very much.
Brilliant. Thank you so much, Elgene, for that great overview of what is really very impressive portfolio of trials activity going on now on campus. So that's really great to hear about. So we've got an opportunity now to ask some questions to our panel. We've had some great questions coming in on the Q&A. Feel free to either put in new questions or to click the thumbs up button for a question that you're interested in having answered. And I thought maybe I'd start by asking Surat about a class of drugs that Liz touched on, but we haven't heard much about yet, which is immunotherapy. I think in the media, people are aware that immunotherapy is this new class of drug that's looking to be very effective in some cancer types, but it hasn't made a major impact yet in breast cancer. So could you tell us a little bit about the latest research in the application of immunotherapy to breast cancer, please?
Yeah, it's a great question. And I think that there is intense interest in the research community on understanding precisely what Liz laid out as to why the therapies that have worked in melanoma and lung cancer have had a very modest, although some impact on breast cancer, particularly triple negative breast cancer, where they've been approved. I think that really the key has been to try to unlock some of what was highlighted, which was understanding what are the cells present in the setting, not just the cancer cell, but the support cells. What are those immune cells and what's restraining them? We really haven't done a great job of characterizing all those cells. So this type of single cell approaches that you have pioneered and that are being developed need to be applied in patient samples with respect to these immunotherapies. And people are doing that, particularly in early stage setting where you can collect a tumor after a patient has been exposed to immunotherapy for 4 months or so, pre and post, and understand what are the immune cells that are present and the support cells, and how did they change with therapy, and what are the signals? Because I think the approach of simply adding the next drug without really understanding what are the restraints on the immune system, how has the cancer evolved, I think is likely to be just as successful as what we've already done, which is just applying the drugs without having carefully known what the sort of breaks are.
Right.
So I think we're still in the sort of understanding why the immune system isn't targeting it, rather than ready for a blockbuster drug.
Do you think with enough research and enough of these insights that you've outlined, immunotherapy will become an important pillar of breast cancer treatment?
Yeah, absolutely. I mean, I think there is no specific reason why the immune system shouldn't recognize cancer cells as foreign, why this isn't a fundamental way in which cancers actually initially evolve, that they somehow escaped immune surveillance in the first place. We just have to understand what was the ways in which that occurred in the case of breast cancer and the different types of breast cancer that are present.
Right, okay, thank you very much. Elgene, we've got a number of questions here about breast cancer risk. And of course, you know, we know there are certain inherited genetic features in some people that put them at very high risk of cancers like breast and ovarian cancer. Could you tell us a little bit about the kind of cancers, well, really, firstly, what's being done in the space of prevention for those women? How can they, you know, what's the latest in managing that risk? And then when they develop cancers, what are those cancers like and how are they being treated?
Yeah, so thanks, Alex. The genetic predisposition to breast cancer has been expanded. Traditionally, we've thought about BRCA1 and BRCA2, but more recently, in the last decade or so, other genes have also emerged as players in this field. So the first thing I would recommend is for anyone with a strong family history to have genetic testing. And should they be at increased risk, the strategies include having prophylactic mastectomy. It's a very personal decision, but that requires taking the breast off so that you would minimize the risk of getting a breast cancer. It's not for everyone, clearly. And for women who decide not to have their breast removed, then we would certainly encourage regular screening above and beyond the standard guidelines for the general population. And there's a particular trial that we're running based on work from the Lindemann lab at the Walter and Eliza Hall Institute in Melbourne, looking at a drug prevention where they receive a drug every 6 months to reduce, or to see if that actually helps reduce the risk of developing a breast cancer.
Right. Amazing. And so we know that one of the effects of those mutations that these women have in their cancer is that it makes their cancers are quite unique and distinct from other breast cancer types. And there's been some evidence that drugs that target the kind of DNA damage that is associated with them have been successful in breast cancer. And so what's the latest, this is maybe a question for Elgene and Surat, you know, what's the latest with that class of drugs, for example, olaparib and similar drugs like that, which have, I think there's been a lot of excitement about their potential for treating these kind of BRCA1 and 2 mutation associated breast cancers.
Surat, do you want to take that?
Actually, in the end, you know, one of the very interesting things that's emerging is using, of course, this approach to target these cancers that have a defect in the normal repair process. And, you know, the class of drugs, the olaparib and others, essentially, you know, accelerate that process and make so many mutations that the cancer cell dies because it's just there's too much damage, so to speak. One of the approaches there is to actually partner that with immunotherapy, because perhaps by making the cancer cell look so foreign through those mutations, you can actually enhance the ability of the immune system to recognize that as foreign. And so that's an approach that's in current clinical trial testing in the early stage, as well as in advanced cancers. And so I would say that's one very interesting area that's moving ahead.
Fascinating. Liz, you told us that some of your work, and obviously many cancer researchers, relies on laboratory models of disease. We can't do everything we need to do in people, right? Because that would be way too risky and unethical. So instead we use these kind of models in the lab. Could you tell us about one of the models that you use and how it really works and the kind of insights you gain from that?
Sure, Alex, I can definitely do that. There are actually a lot of different models we use, and we're trying to make those models more and more relatable to the human disease as we go forward. So at the beginning, when models were first used, it was cells that had been taken from a breast cancer and grown in a plastic dish in the lab. And a lot of information was got from that type of experiment. But now some of the models that we use are models, and Elgene's group particularly uses these models. We use what are called patient-derived xenograft models, where a piece of tumour is taken from a patient, and then that piece of tumour is grown in a mouse, implanted in its mammary, in the mammary gland of mouse. And that type of model is extremely powerful. It allows us to test the effect of a drug in a proper biological system and look at the effects of how that drug is metabolized in an animal and then gets to a cancer site and actually causes differences to that cancer, hopefully to regress the cancer or stop it spreading through the animal. It's a big decision to do experiments in animals, but often those sorts of experiments are preceded by a lot of work with those simpler models which we do in a dish. So we thus in that way develop a really strong understanding of what we could achieve by doing those type of patient-derived xenograft models, and then do a test which will see us take a new treatment forward to clinical trial.
Right, so I guess what you're telling us is that those patient-derived xenograft models act almost as an avatar for a patient, and that you can then run almost mini clinical trials in those models. Is that true?
Yes, absolutely. So we often use groups of animals, like a small group of animals, to test drugs on to mimic that diversity that we see in people. So we might have a small group of like 5 to 10 animals to trial a new drug on. And in that way, we can see that the drug is in fact working in 5 to 10 different biological systems. And that gives us a lot more confidence taking drugs forward to actually put them into humans.
Right. Thanks very much, Liz. So Kate, I guess from a mouse in the lab to you, a willing participant in a clinical trial, you know, I think we talked a lot about your medical management, but obviously a diagnosis and treatment of cancer is an enormous emotional journey as well. And not just emotional, that there are huge logistics to deal with in your personal life. And so what kind of support groups have you relied on? Have you found, for example, networks of patient, you know, people in a similar situation to yourself that you've been able to connect with and learn from?
I did. I have a very close friend who had a very similar experience, similar diagnosis to me. And that was, I think she was diagnosed probably about 4 or 5 years ago. So she's been a real great source for me. I did join the Breast Cancer Network of Australia, and I must admit I'm not an active participant, but I do go onto their site quite a lot and have a look to see what's going on. And so it's, yeah, I found it can be in the beginning quite overwhelming because, as we've heard, there are so many women who have got breast cancer, and you also, I'm sure people who are listening who have had a cancer diagnosis, you suddenly hear about everyone's distant relative cousin who has cancer, and they all want to give you advice about everything. And I got very good advice in the very beginning, was you have to just realize that what people are saying to you, it all comes from a good place, it comes from the heart, but it can get a bit overwhelming just hearing it all. But also I just had incredible support, particularly from my workplace as well, because when I found out about the diagnosis, it was a phone call whilst I was at work. So they were the first people to hear it, and they've been, so work has been incredibly supportive, and I think that's has helped me a lot, and they've given me the time that I needed to take off work. And I started back at work this year when I was doing the radiotherapy, and yeah, they've kind of like, you know, whatever you need, just let us know. So that's been great. And, you know, there are so many groups out there. I'm booked in for reconstruction quite soon, and on Facebook, you know, I found a group that is that have had the same operation as I have. So they've been like just getting like even like little hints about, you know, what to take to hospital and things like that. It's great. So yeah, and you know, family. So just yeah, I think that's really important. And again, I think that must be very hard with COVID for people who are at that earlier stage going through, because having that, you know, face-to-face interaction with people is, yeah, it's just, and you just, that feeling that you're not alone which is a terrible thing to say because you know that so many people are going through the same, you know, experience that you're going through. But there is so much help out there. If you need it, you can, you know, find the groups and be very active in them if that helps you get through.
Well, I think that's a really wonderful positive note to end on. This is a wonderful conversation. I'm really enjoying it. I could chat to you all day, but we've reached the end of our symposium. And I really want to thank you all for your contribution. It's been really outstanding. We hope you have a lovely day. I want to particularly thank Estee Lauder and Emerentia for joining us today, for their support of the symposium, but also for the support of Sarah's research proposal. And if you join us next year, I'm sure you'll be hearing the exciting early results from that research. So with that, I'd just like to wish you all a wonderful day, and thank you very much for joining us.